Hello everyone, in this video I'll be demonstrating on how we can upload our region to SRA. SRA stands for Sequence Read Archive. It is a site in NCBI where we usually store unassembled reads. Basically, these are the reads that we usually get after sequencing, depending on the sequencing method used. So first of all, we have to download an application that will enable the connection between our computer and NCBI. So we'll go to Google. My network is low, I don't know why. Okay, here we are. Go to Google and type Aspera Connect. Download. That's the application that we're gonna use to connect our computer into NCBI. And then we click download Aspera Connect. After that, we have to download Aspera Connect. If you are using Windows, just click download. But if you are using MacBook Free, you have to click macOS. Then if you are using Linux, you have to click Linux so that you can be able to download an application that is suitable for your operating system. I've already downloaded this application before, so I won't be downloading it. But you can download it, just like click and then downloading and start. And then after that, in your downloads, you're gonna find an application file like this one. So you have to install it by double clicking it. But me, I've already installed one, but to install it, you just double click it. And unfortunately for me, I can't install it now because I've already installed one. So just cancel this one. And then after successfully installing the IBM Aspera Connect, you're supposed to have a pop-up window like this one to show that you are ready for uploading our data into SRA. So I'll quickly minimize this one and then we'll go to the next step, which is actually submitting the data. Go back to Chrome again, and in this case we have to go to NCBI. So I'll click Google and then boom, here we are, and then I'll have to type NCBI and then click NCBI again, and then here we go. I click submit because we're submitting rates towards to NCBI, so I'll click like submit. And then it will ask you, what do you want to submit? In our case, we are submitting rates to SRA, so I'll click SRA, and then it will tell us what kind of rates is accepted by SRA. SRA accepts unassembled rates from high throughput sequencing platforms. Submitted data files should generally be minimally processed and include PEP-based quality scores. And SRA accepts different file formats depending on the sequencing method used. For example, in this tutorial I used FASTQ formats because for sequencing I used Illumina sequencing, and Illumina sequencing most of the time it produces FASTQ formats. But like there are other file formats that are acceptable, like BEM formats, blah blah blah, depending on the sequencing method used. And then we'll continue, and then we'll click submit. And then I will click like new submission because I don't have a submission yet. New submission. And then boom, here we are. Here I have to click like to fill in my details, but because I've already logged in as Alsenia in NCBI, the website has already detected for me my details like first name Lungelo, last name Kanile, email Lungelo, blah blah blah. So now, and here you have to fill in your organization. If you're having a research organization, you fill in your research details and everything, the department, your lab, blah blah blah. And then here you click the address. Me, I'm currently in this address, so I just filled this address. So, and then after that you can click continue. And then you're going to step 2. Step 2 is general information about the project. here it's asking like, did you already register a bio project for this research? In my case, no, I don't have a bio project for this research, so I'll click no. But if you have one, you will click yes, and then you have to fill like this number. As we know that like in NCBI, bio projects usually start with PRG, and then you have to fill in your number of the bio project so that these rates can correspond with the bio project you submitted. But in my case, I'll just click no, and then after that we go into the next step. Did we register? No, we didn't register this one, so I just click no. And then when should the submission be released to the public? Now I'm not sure yet, so I'll just like say release immediately following processes, because if I click like release on a specific date, I have to fill a date, and but currently I'm not sure, so I'll just like click release immediately following processing, and then I'll click continue. And then now we go to project information. Here you just like write the information about the research project that you are currently doing, where you used layer rates that you want to upload on this one. For example, I'll just be giving an example. So here I'll just like say transcriptome analysis. Let me just say transcriptome analysis of malus domestica. Those who are specializing on plants will know that malus domestica is in April, so I just click that one. And then public description, I can write anything for the sake of this tutorial, but when you're uploading your data, you're supposed to write the correct information. For example, I will write like in this research we aimed at I'll just leave it like this. I know it's not the right thing, but I'll just leave it there. And then go to relevance. And the relevance, it will depend on the type of research you're doing. But now since I've used April, I'll just like click agricultural. But let's say like we're doing like a transcriptome analysis of an animal, or transcriptome analysis of another thing. You can even click like medical. But if you're doing transcriptome analysis, or your research is based on industrial, you click industrial. If it's environment, if it's evolution, you base it in it depends on the type of research that you're currently doing. But me, I'll just like click agricultural in this case. And then is your project no? Yeah, like most of the time you have to click no. Yes, it's not very common as stated by the website. And then as you don't have external links, we click this one. Grants, don't have grants for this, we click this one, and then you can just click continue. But if you have grants, you can add grants here by clicking add grants. That's where you're gonna be right at click grants number, search by tag, blah blah blah, and that's it. But as we don't have grants, so just like click continue. Here we go, but my Wi-Fi is a bit slow. Boom, here we are. Now we have to fill like biosample type. Here biosample type, just like explaining what type of data that you're uploading to NCBI. For example, if you're uploading model organism animal, you click model organism. But this step is very important, because if you press the wrong option or you click the wrong option, it's going to affect you in the upcoming steps. You won't be able to proceed with the application if you made a mistake, because in the upcoming steps you also have to include the name of the name of the organism that we are dealing with, so the website is able to trace back and check if you click the right option here. For example, if you can click pathogens here, and then you go to that site, and then you click an organism like Malus domestica, you won't be able we won't be able to proceed with the application because it will be wrong. So in my case, I will click plants, because they put it in plants. Everyone knows that. So I click like plants, and then I click continue after that. Here we go. A bit slow. Boom, here we say biosymbol attribute. Here are two ways of doing these things. You can either like you use built-in table or you upload a file using Excel or text format. But in this case, I'll use built-in table edit. But you can also like prepare an Excel and then just click upload, boom, which is like I think it's the easiest way. But in this case, I'll just like use built-in edit table. And then I have to fill like this table. For example, assembly name, I can just click my name. Assembly text is not important. The important parts are the one that like I'm marked with a star. Organism, yes, go like say like loose domestic. I think I didn't misspell the. I said that you can just click like non-applicable. Type in cultivar. I think we both know what a cultivar is, but like if you don't know what a cultivar is, you can click information cultivated variety of plants. But you can even skip this one, it's not important. Then just click ecotype. Ecotype, the population within a given species displaying genetically based phenotypic traits that reflects adaptation to local habitat. In this case, I think it is wise to specify the area where you took your samples from. In my case, I'll just like click check and then age adult stage. You can also click like in age you have to specify the number of years, not adult stage. But I'll skip this one, it's not important. And then development stage, adult for me because I took my samples when they are under like the development when they were adults already. So now we'll just proceed to the next thing, which is geographical location. Geographical location, we'll click specifically where you took your samples from. In our case, I'll just type like Turkey, and then if you want to be more specific, like on the area, you can include semicolons, and then me, I took my data from Kayseri. Kayseri is a town in Turkey. Tish, in this example I used a plant, so either like you use leaves, roots, stem. Like in my case, we use leaves to get the data, so I click leaves. Then the other things are not important, you can just keep them, and then we'll click continue. So hopefully we filled everything right, because if we made a mistake, boom, luckily we did. So now we have to click SRA metadata. SRA metadata, you can also like upload it in two ways. First way is to use built-in table data editor, where you have to write and fill in the data online on the site like we'll do now. And then the second one is to upload a file using Excel or text format. In the second one, you just prepare the file before, and then when you're trying to upload the data, you just click upload. I think it's the easiest way, the second one, but in our case we use the first one. We just built-in table editor. So sample name, I think I used Lungelo before, so I won't change it. Put Lungelo. Sample ID. These IDs usually like they usually come with the sequence reads. So I just like put one like here, for example. Title, I think we did like transcriptome analysis of mouse domestic, if I'm not mistaken. Then say library source. This is the most important part. Okay. Then after that, you have to say library strategy. That's it. Libraries that you click, you select the most appropriate one. If you did like whole genome sequencing, you click like whole genome sequencing. But in our case, there are a lot of different options. You click like the right option for your research or for your data. In my case, I'll click RNA sequencing. So boom, here we go. Library source, it's a lot. You can click like genomic, transcriptome, single cell, metagenomic, depending on the data or the file that you want to upload. But in my case, it's transcriptomic. As you said, like my topic is transcriptome analysis of malus domestica, so it's transcriptomic. But like in other cases, for example, if you do like maybe a whole genome sequencing, I think twice to click like genomic. But if you're doing like viruses, then you have to click like viral RNA. So I'll click transcriptomic, and then library selection, different library selection. Maybe either you use like PCR, random PCR, real-time PCR. You click the one that you used for your research. In my case, I just use PCR. And library layout. There are two types of library layout. There is singly, and then there is paired end. Where single is just like in one direction, and then paired end you just got like a reverse and forward read. So I used paired end. And then platform, depending on the sequencing method you use. Platform is the sequencing method you use. As we use the Illumina sequence, so we have to click Illumina. If you use like capillary, complete genomics, ion tolerant, you will click the other option. But like in our case, we use Illumina. And then here, as you know that Illumina has got a lot of models, like this 2500 X10 if I'm not mistaken, Nova's like 6000 if I'm not mistaken. So we click the right model you use for your data. In my case, I use like Lumen 6000, so I'll take this one. And then design description. Here it's just like a method of briefly describing about the methodology. So I'll just like write I'll just like write one line for the sake of this video. For example, like RNA was extracted from the leaves, let me say fresh leaves, using the setup method. Basically, you just have to write a summary of your methodology. And then file type. There are different types of file depending on the sequencing method used. If you used like Illumina, 90% you will get FASTQ files. So in my case, I will use FASTQ files, but like other different sequencing methods, they have different file formats like BEM files, PacBio, Complete Genomic Code, Oxford Nanopore. So depending on the sequencing methods used, you have to click the right file format that we have. In my case, I have FASTQ file format. I click FASTQ, and then in the file name, in this file name you have to make sure that you write the appropriate name or the same name as the file that you want to upload, or the reads that you want to upload. For example, in our case, I want to upload academics genome start. Boom. Why is my folder, by the way? No. In my case, I want to upload like these two types of these two rates, and it's just one rate, by the way, because like this is the forward rate and then this is the reverse rate. But since it's paired type of rates, I have to upload both of them. So I have to make sure that I write the appropriate weight. So to avoid this, I just like right click and try to rename them, and then like this, and then I'll like Control-C to copy the rename so that I cannot make a mistake. And then I'll go back to my file name, and then I'll just copy and paste That's it. And because in the in where is this thing? Let's go back. Here in the library layout, it was paired end. So in paired end, we have to make sure that we upload two files, the forward one and the reverse one. You can't be able to proceed if you have uploaded just one file. You have to upload two files. In single end, you can proceed with one file, but in pair end, you have to upload two files. So here is my forward one, and usually in Illumina sequencing, the forward one is usually R1 and the reverse one is usually R2. So now I have to come and click and upload R2. I'll just do the same thing so that I can get the exact name to avoid making mistakes, because if I make mistakes I won't be able to download my file. And then I'll paste it here. Here we go. Now we have our files. We can continue. We click continue and wait. If we did a mistake, we won't be able to upload our data, but if we didn't, we're gonna be able to move towards the next step. Let's wait and see. Still processing. Let's wait and see. I'm sorry, my network is having problems. Let's wait and see. I hope I didn't make a mistake. Hopefully, because we have to proceed to step 7. If we didn't make a mistake, we have to proceed to step 7. So here we go. We didn't make a mistake. Now we have to proceed to step 7, which is actually uploading the files towards NCBI. So here they ask you, how do you want to provide files for the submission? You will click different options depending on how you want to upload your files. I think I will elaborate on the other methods on upcoming videos, but in this one I'll just use Aspera Connect plugin, the one that we downloaded before at the start of this tutorial. So here we go. Click this one and then select choose file and drop them in. So I'll go to choose file, simple like that. Choose file, and you say like yes. It will allow access to your computer, because we've already downloaded, like we've already installed IBM Aspera Connect. So we have to give it access to our computer via Aspera Connect. So we click yes, then wait. Supposed to pop up something. Boom, here we are. So now we can be able to select the right file that we want to upload. So here we go. It's master D. This is the forward rate for my data. So I'll click it and then I'll say open. This is open, but it's just that I'm using a Turkish computer. So I'll click Arch, which is open, and then boom, it is loading. And then like you must give permission. So yeah, I'm giving permission for your computer to work with ICBN, NCBI. And then since we have to upload two files, the forward one and the reverse one, so we'll just continue and say choose file again. Okay, which means that we can only be able to upload one file at a time. So we'll just wait for this one to finish. But it's gonna take time, but I will come back to continue when it finishes uploading.